So I guess I'll introduce myself first. So my name's David. As I said before, I'm joint between Ben Cosgrove's lab and Ewan de Vlaming's lab. I'm a third year, I think. Time is hard to keep track of in the time of COVID, but third year. Yeah, so I work on single cell RNA sequencing on kind of a few different areas of that. So because we're engineers, always thinking about methods development and finding new ways to measure things. And then on the biomedical side of things, in mostly with Ben Cosgrove's lab, we study muscle stem cells and use single cell RNA sequencing tools to tease out the dynamics of muscle stem cell differentiation and try to find new ways that we can culture these cells ex vivo to develop stem cell therapies. So yeah, Ben, do you want to maybe introduce yourself next?
Sure. My name's Ben Grodner. I'm also a third year in Ewan de Vlaming's lab. Sort of a different side of the lab. We're doing method development for technologies to spatially map microbiomes. So microbial communities in the human body are complex. They're composed of many different taxa, and they have intricate spatial structure. And most of the tools that we currently leverage in studying microbiomes destroy that spatial information. And so we're working on imaging and biomolecular staining techniques that allow us to multiplex readout of these microbial communities and study these complex systems as they exist in place. And we're working in a couple different systems in mouse gut and then human oral plaque. We're looking at some colorectal cancer models. And yeah, so I guess, I don't know, how should we proceed, David? We could show some slides or we could just move straight to questions.
Yeah, maybe let's do we'll vote thumbs up or thumbs down if you want to hear about, like, maybe if you want to see us give like two or three slides of our research, thumbs up, or if you want to go straight to questions, do thumbs down. That way you don't have to talk.
Too many slides today. It's fine.
All right, I think we got research. Do you want to start?
Sure. Can you enable sharing?
Every time. Should be good to go.
Okay. So, mapping the microbiome. Again, this is Ewan de Vlaming's lab. This is a look at sort of the outputs that we're getting with this new method that we've developed. We're segmenting single cells and assigning taxonomic identity to these densely packed microbial communities. This is a sort of a mixed image of human oral plaque, the stuff that grows on your teeth near your gingiva. So this is all motivated. This sort of summarizes some of the motivation for the development of these techniques. We are interested in quantifying and comparing across systems, these highly complex systems, and to do that we need to multiplex our ability to do in situ phylogenetics. So yeah, basically the technique that we're developing follows on a bunch of microbial research going way back in time to the 1600s, when this guy Antony van Leeuwenhoek first looked with a good enough microscope and discovered that bacteria exist. And since that time, microbial research has gone through a couple different trends. The first trend was a reductionist trend, where you're looking at more and more detailed view of bacteria, studying bacterial, individual strains, isolating them, isolating genes, isolating proteins, and discovering, interaction networks. And then as we've, developed better and better technology with better computational approaches, and especially the advent of sequencing, we've been moving back towards studying bacterial species in multi-species communities as sort of a holistic approach to microbiology is coming back. And so bulk sequencing of 16S rRNA is sort of the main tool in these microbial community studies. What you do is you extract a community from its environment, grind it up, and amplify the 16S ribosomal RNA gene. This gene allows you to determine the evolutionary distance between all of these different taxa, and you can get a compositional readout of the microbial community. And you can, for example, in this paper, determine correlations between certain microbial compositions and, for example, response to a drug. The issue with this measurement is that it's sort of one-dimensional. You only get compositional information, and you would destroy all of your spatial relationships, as I mentioned. So this is the Lammerding Lab. Most of what I'll show is spearheaded by Hao Shi. He recently defended, and as well as we collaborate quite a bit with the Brito Lab, Ilana Brito and Warren Zipfel. And then all of the imaging stuff is done downstairs at the Cornell BRC. So these are all sort of, great resources that we have available. And so this technique is called hyperfish, high phylogenetic resolution fluorescence in situ hybridization. So lots of different phyla are present in these species, and so we're creating high phylogenetic resolution maps of them, and we're using fluorescence in situ hybridization. This is a technique where you're staining using Watson-Crick base pairing of DNA molecules to create specificity in your stains. And with this approach, you can identify and locate the bacteria in the community. This is another look at oral plaque. And so basically what we're doing is we're creating combinatorial barcodes, fluorescent combinatorial barcodes to read out individual bacteria, as well as pretty advanced segmentation algorithms and some clever probe design as well. This is a look at how the technique works. Again, you're labeling the ribosomal RNA, which differentiates species, with these single-stranded oligonucleotide probes. The probes are then hybridized with fluorophores that create these fluorescent barcodes. The fluorescent barcodes are imaged on a confocal microscope with spectral readout. So each pixel has 95 spectral dimensions, which create the spectral curve. We then segment the cells and can deconvolute the curves to determine which fluorophores are present and barcode the species appropriately. You can measure physical relationships such as orientation in these highly structured biofilms. You can identify consortia that commonly group together. For example, this three-member consortium that we identify. This is a three-dimensional image of what those typical consortia look like. And then you can also, with the high multiplexity of hyperfish, identify rare species or lowly abundant species, which are can in fact be important to community structure. You think of like keystone species that have an outsized effect on community structure despite their low abundance. And then this is another sort of interesting analysis that we can do. Like once we identify once we identify a bacteria, we can also we can combine with other imaging measurements. This is a super resolution microscopy that allows us to look at the ribosomal RNA distribution within a bacterial cell. And we can see how different species of bacteria take different approaches to how they distribute their ribosomes. The other thing we can do with the single cell segmentation is look at physical relationships and quantify those physical relationships. So for example, Bacteroides tend to associate with themselves. So as you move radially away from a Bacteroides cell, you're very likely to find another one very close by, whereas Aspergillus cells are sort of homogeneously distributed throughout the biofilm. This is a look at an image of mouse gut. There's a mucosal layer, and then the host cells, and then this biofilm layer. is how intestinal microbiota is structured. And you can also look at pairwise correlations. This is a look at the differential spatial association between antibiotics treated and non-antibiotics treated mouse. And so you get sort of these changes in the microbiome structure, how species associate with each other, depending on whether or not you've applied antibiotics to these mice. And so, yeah, with that, I'll try to move through that quickly. So, and then again, we can ask questions once we turn off the recording, I guess. I don't know, David, did you?
Yeah, maybe I'll go real quick, and we'll try to leave the last 10 or so minutes for questions. Let's see here. So, alrighty. So as I said before, I work in Ben Cosgrove's lab and Ewan's lab, and we use single-cell RNA sequencing to study muscle stem cells and other types of cells in skeletal muscle. So I won't go into what single-cell RNA sequencing is. I think the name says it all. Essentially, you isolate single cells and sequence the RNA inside of them. And the system that we're interested in is really this process of muscle stem cell differentiation. So on the periphery of myofibers, which are the cells inside skeletal muscle that allow you to flex, is where these satellite cells or muscle stem cells sit, and they follow this process of activation and then self-renewal, proliferation, differentiation, and then they fuse and form new or larger myofibers. And it's a pretty complicated process. When you age, of course, it degenerates and people lose the ability to heal as efficiently. So we're interested in why, essentially. So a couple years ago, before I actually joined the lab, well, I guess it was published after I joined the lab, but the Cosgrove Lab's first endeavor with single-cell RNA sequencing generated about 60,000 cells, single cells, transcriptomes. And in the plot I'm showing here is isolated muscle stem cells and all the myogenic cells. And we use a myriad of computational tools to order these cells along the process of myogenic differentiation. So as you can see here, the muscle stem cells are here, and they're colored by cell type, and also by, we use an injury response model where we injure mice and then collect cells at certain time points after the injury. And so they're colored by injury response time point and also cell type. So you have the muscle stem cells here, and you can see process of differentiation, proliferation, and then maturation, commitment, and fusion. So when I joined the lab, I saw this kind of data, and I thought, well, that's really interesting, but there are a lot of gaps in this process. So like, what's happening in this area, and what's going on? Most of the ways that we look at muscle stem cells is by really actually sparsely expressed genes, so transcription factors and surface markers for the most part. And so what I'm showing here is a plot from another paper. These are a couple really important genes in myogenesis, and along the x-axis is the number of transcripts per muscle stem cell. So you can see these transcription factors, which are the first three, are present in 10 molecules per cell, which means when you're looking at cells that account for only 1% of the total number of cells, and you're looking at these 10 molecules account for essentially 0% of the molecules inside each cell, you need really sensitive methods. And so the approach I decided to take was, rather than trying to improve the sensitivity of single-cell RNA sequencing, why not just take more measurements? So I gathered a lot of data. So here I'm showing on the left our data plus several other labs' single-cell RNA sequencing data from mouse skeletal muscle, and using a tool called UMAP to take essentially all 25,000 genes and project them into just 2 dimensions. So on the left, the cells are colored by the original source of the data, the people who generated it. And on the right, I've clustered the cells and used a bioinformatic tool to correct for differences between batches. And here they're colored by cell type. So you can see, there's like endothelial cells. And because we have so many cells, we can distinguish differences between like venous endothelial cells, arterial, capillary, things like that. Lots of different immune cells, fibroblasts. These are fibrogenic, adipogenic progenitor cells, kind of like mesenchymal stem cells in muscle. And really most interestingly for me, we got all of these myogenic cells here. And so the next thing I did was I took these myogenic cells and used a different tool to order them along this process of myogenesis. And so what we're seeing here, essentially the x-axis is this process of myogenesis where you have the stem cells here and they differentiate, and you have the mature cells here. And what I did is I just bend everything to make it a little bit easier to analyze. And then, so this is a panel of known muscle genes. And so you can see these waves of differentiation going from left to right. Essentially this wave of different genes getting turned on. And with this kind of data, we can begin to explore, new genes that people haven't studied before. And so what you can do is, what I did was perform a bunch of differential gene expression analysis and pull out surface markers here on the left. So everything below the dotted line is a new, never been looked at surface marker that marks these intermediate cells that are super rare in these data. And on the right is transcription factors. And then the next thing that we did was take this data and alongside it, we generated spatial RNA sequencing data. So each of these little dots is a barcoded spot on a slide. And we use the barcoded spots to capture RNAs, and then we can sequence those RNAs and map them back on to where the spots were, and we get these spatial pictures of total transcriptome expression, right? So you have the H&E stains, which is just looking kind of at the general tissue, what the tissue looks like, and you can see this is 2 days after injury, 5 days after injury, and 7 days after injury, so you can see all this immune infiltration in purple. These are a bunch of a bunch of myogenic genes that I had shown before, but kind of here shown in space. And with, so remembering back to that big kind of compendium of single cell data, we use another informatic tool called Bayes Prism to tease apart each of these individual spots into the constituent single cells, because each of these spots is actually more of a bulk experiment that contains about 10 cells. And essentially, I can find in each one of these spots what percentage of the molecules are derived from each of these cell types. And you can begin to, rather than just looking at kind of broad gene expression, you can begin to see patterns of how muscle stem cells and other myogenic populations are distributed across the tissue, across injury. And then we could do other things like look at co-occurrence of each of these cell types with each other and see over time which cells tend to co-occur more often with each other, which kind of implies that they have some sort of relationship that's important to this process of myogenesis. But yeah, I think with that, because that's my last slide, I'll stop, and maybe now we can stop the recording and switch over to some questions.